Bow your heads, please.
Thank you, Lord, for this day that you've given us. I pray that we all did well on our tests and that we just learn from this class. Thank you for Ms. Frady's and just teach us. Amen.
Amen. Thank you. And a little heads up, the time to pray for a test is not after you've taken it. Okay? And it's interesting. I have to tell this to all my students. I know. But the time to pray about a test is if you pray while you're studying. That's really helpful. Pray before you study. Lord, remember the Holy Spirit's our teacher. So you pray before you study, and Lord, please help me to understand this, to grasp the things you would have me to, so I can use them to your honor and your glory. And then right before you take the test, that's really the time to pray hard.
Or you could just blindly.
Well, in college, I had a standard test prayer. Okay? And you have to understand, when I was in college, everything was on the quarter system, which means most of my classes, you had 3 tests that made your whole grade up. And so they were 3-hour tests. They were done on usually Saturdays. You know what I'm saying? And so each test was a huge chunk. You blow it, you're kind of done, all right? And so my standard test was, and I didn't want to waste time because you didn't have much time, so my standard test was, Lord, please help me to remember everything I've learned, studied, heard, and let me take good educated guesses on the rest. And that was my standard prayer, okay? Because I honestly, a lot of tests, if you knew the information, you could figure out the answers, but you didn't necessarily just know the stuff just because you had studied everything you were supposed to. One of my favorite teachers, his tests were a learning experience. And I had 102 average in his class because I thought that was so cool. Because if you studied what you were supposed to, you could figure out the answers, but it was like being a detective. And so that was my micro teacher at Auburn, Mr. Wilt, and I just loved him. I thought he was so great. But his tests were really a learning experience. So anyway, okay, let's go ahead and start on the test. And David, would you start us, please, sir, by telling us what a heterotroph is?
An organism requiring organic compounds for its principal source of food.
Wow, yeah, that sounds good. Something that eats some other source for food, it doesn't make its own food. Very good. That's good, Jonathan. That's totally right. Something about it getting its food from some other source. It does not make its own food. Anything that says that is fine. Thank you. How about mutation, please, sir?
Is that B?
That's B, yes, sir. Hang on just a sec. Guys, everything you do, talk-wise, they're hearing you. Just so you know. Okay, go ahead.
That's a big change.
Good, a big change. Thank you, sir. And so you could have put a copying error. You could have put a big change because it's going to look very different. Anything like that. Okay? Thank you very much, Connor. Jacqueline, how about a theory?
A hypothesis that has been tested successfully.
Very good. So it's a well-tested hypothesis, or you could have even said a well-tested educated guess. Very good. In case you didn't know how to spell hypothesis. I can relate to that. I can do it now, though. That one at least is phonetic, isn't it? You can sound that one out. How about photosynthesis?
Converting light into energy.
Okay, actually it's converting light into food. But I'll take energy, leave it. You know why? Because why do we use the food for energy? But just put food over the word energy so you know it's for food too.
Right.
In the book, did it say that?
I think so.
No, go ahead.
But you can't take energy and then convert it to energy.
Right, exactly. But what I think she's saying is you're taking one form of energy and converting it into living energy, and that's what we do with food. So that's the reason I'm going to let her slide on it, because I think I know what she's trying to say. Right? Okay. But I agree with you, and that's why I said right over it food, but why do we eat food? For energy, so. Okay, so we're in good shape. All right, so Dominic, will you please tell us what a prokaryotic cell is?
A cell with no membrane-bound organelles.
Good. Should have had no membrane-bound organelles. That's good. Thank you. And then Eva, how about species?
Species are organisms that are able to reproduce and their children are able to.
Very good. Something about they can reproduce and their offspring can reproduce. Very good. Thank you. And so, Kelly, we're up to number 2. Would you read that for us, please?
I don't have the question.
Okay. It says an organism is classified as a decomposer. Is it an autotroph or a heterotroph?
Heterotroph.
Good. It's a heterotroph. And if you guys can, bring your tests for when we're going to grade them so that we can read through them together, okay? Thank you. You didn't know. I know that. Audrey, do you have a test that she can read? Okay, number 3 is, a tiger develops lockjaw and can no longer open its mouth. Which of the 4 life functions will it not be able to perform?
I wasn't able to answer that.
Okay, wait. Shh, don't say anything. Okay, now I want you to think for a minute, because I know the way he asked this is kind of hard to know what he's looking for, but you could still answer this question. Think about the 5 criteria for life, and he gave 4 criteria for life in the book. Okay, what were the, do you remember what they are?
Needs to have DNA, needs to be able to reproduce, needs to have cells.
Okay, cells is one of them.
Okay.
One of them is if it responds to stimuli, right?
Senses.
Right, and then the other one is kind of what photosynthesis was about. What was that about?
To be able to have, how do you pronounce it, metabolism?
Good. Metabolism. Good. Metabolism. Now look at that question, and he wants you to answer it with one of those things.
Oh, metabolism.
Thank you. Write it down. Okay. Don't want you to miss that one. And it is, the way he asks it is odd, and I understand that. And what he was looking for is one of those things that were lacking. Yes, Connor.
For that one, I didn't really know, but I just went with logistic growth because I thought it was like around it.
Logistic growth because logistic growth is limited because of resources. I'll give you half for it, Connor. Okay? Yes, ma'am.
I put it won't be able to convert energy from its surroundings into energy from
That's metabolism. You've just
So I just should have put
no, honey. You just explained metabolism. It's all good.
I think I got that one wrong. I put it couldn't open its mouth.
Yeah. Okay. But you tried. At least that's good, Daniel. Okay. All right. So we're up to number 4. Could you, Geneva, could you lend Sabrina your test so she can read number 4, please? Thank you.
In laboratory studies, organisms from group A produce offspring with subtle differences as compared to the parents. Organisms from group B produce offspring that are identical to the parents, and organisms from group C produce offspring with marked differences as compared to the parents. Which group reproduces asexually? Which group produces sexually? Which group is experiencing mutations?
Thank you.
So I put group A produces sexually.
Mm-hmm.
Group B produces asexually,
Mhm.
and then Group C is experiencing mutations.
Absolutely. Thank you. And if you happen to just write it on there where it was asking it, it would be asexually would be B, sexually would be A, and then mutation would be C. Absolutely. You did it absolutely right. I just wanted, in case somebody just wrote down the answers in that order. Okay, great. Thank you. And then Geneva, would you read number 5 for us, please?
A biologist studies 2 organisms from the same family. She then studies 2 organisms from the same genus in that family. In which case do you expect the most similarities between organisms? I'm not sure I got this one right, but species?
Okay, don't say anything, because we're going to still look at this. They didn't give you the choice of species. They gave you the choice of family and genus.
Oh, okay.
So which one would you pick?
Genus.
Good girl. Genus. Absolutely. It's okay. Just write genus. You got it. That's good. So they were looking for which one within those 2 would be the closest together. Thank you. Number 5. And that goes for you guys. If you messed up on the test and then you are getting it as we do it, that's fine too. You don't have to count it off. Or you could always take the test over if your mom wants you to do it that way. That's fine. Okay, Jonathan. Let's see. Yes, sir, please.
A biologist wishes to study how two remarkably different plants react to the same changes in their surroundings. Should he choose organisms from different phyla or different orders within the kingdom Plantae?
Good, and it's phyla.
Phyla.
Yeah, you know how they're weird in the Latin thing. Okay, phyla or Plantae, and we're looking for the biggest differences. So which one would it be?
Phyla?
Good, the phyla, because it's right up at the top. Kingdom phylum, so that's, really big. Okay, great, and we're looking for the biggest differences. Number 7, please.
I don't have the book.
Okay, it's okay. It's okay. Okay, you're going to just share it? Thank you very much, Andrew. Number 7.
What did the law of spontaneous generation survive for so many years? Why? Sorry.
It's okay.
Because of Aristotle's theory, they didn't want to go against him, so they just thought he was correct, so they just stuck with him.
Okay, so that's a good answer. You could have put that they misread the experiments. You could have put that they had faulty experiments. You could have put that's the same idea. They had flawed experiments. You could have put that they misinterpreted the experiments. What'd you have, Audrey?
I put because of many flawed experiments.
That's flawed experiments. There you go. Dominic?
And I said because it was never properly tested.
That would be because of the flawed experiments. Yes, absolutely. So anything that's referring back to the fact that they didn't really collect the data correctly or interpret the data correctly would be right. Eva?
I said that they kept pushing the theory into the realm of the unknown.
Yeah.
I'll accept that. Yeah, that's good. That's good. They did push it into the realm of the unknown. Yes.
Okay, so I put, they tested it with stupid experience.
Yes, but we're not supposed to use the stupid word. Yes, okay.
And also, I also put with the church influence too.
Yes, but let's not blame the church. Let's blame the people. But that all had, it was a politically correct thing. And at the time, the church was the government, not the church, per se.
Yeah.
So that was tough. Yeah, it made it hard. Yes.
I just said because of Aristotle's theory.
Because of Aristotle's theory. I'll give you half for it, okay? I know, because just saying that, you're not really telling what happened. And there was a lot that was happening, okay? Thanks, Kelly. All right, on number 8, you guys were supposed to use the biological key to try to solve that, but on the first one, you didn't have enough information to take it all the way. Everybody on number 8A should have plantae. That much you could do. And apparently you could all do anthophyta. So that much you could do. But class dicotyledonea, you guys didn't know what netted or parallel venation is, and so if you missed that one, let it go. If you got it, let it go. I don't care either way. The point is you didn't have the information to do that part, so just let that one go. So you should have had A, plantae, B, anthophyta. Okay, and then on the second part, it's A, plantae, and anthophyta, sorry. B is Animalia, Chordata, and class Aves. Okay, so kingdom Animalia, phylum Chordata, class Aves. And A was kingdom Plantae, phylum Anthophyta, and don't worry about the class. Yes, David?
What if you did put the class?
I'm not going to go either way. Just let it go. Okay? Yes?
So if you missed C on A.
You're just ignoring it. You guys didn't have the information to answer that question yet, and I know that because I know it's in the last 2 chapters of the book. Yeah, so he just, it's hard writing tests. I know, I've done it before, and everybody says my tests are killer because it's very hard to know what somebody really should know. So we're going to cut them all sorts of grace on this, okay? And I'm going to cut you grace. You didn't know that, so we're going to let it go. Yes, and then we break into song, let it go, let it go. Yes, okay, go ahead.
I just put
You put what?
I put class A's. That's all I put.
Aves.
Yeah. I didn't put the,
you didn't put the first. Okay, but you got to the class. Okay, that's fine. That's fine. No, but you're supposed to write them down. But you had to get there to get that answer. So that's fine. All right. Are we ready? So, Corinne, you're going to read number 9 for us, please, from that book. Because Andrew's such a nice guy. Oh, that's Daniel's book. Thank you, Daniel.
An organism is multicellular decomposer with eukaryotic cells. To what kingdom does it belong? I said fungi.
Good. Very good. Thank you. And Daniel, how about 10 for us, please?
Okay. An organism is a multicellular autotroph with eukaryotic cells. To what kingdom does it belong? I put plantae.
Good. Plantae. Thank you. And Andrew, number 11, please.
An organism has a single
prokaryotic
cell.
There you go.
To what kingdom does it belong? Monera.
Very good. Thank you. And David, we're back to you.
If we were using the 3-domain system of classification, in which domain would an organism made of single eukaryotic cell be classified? I wrote Protista.
Okay, you know what? I'm going to give you a half for that because I had several people do that, and that would be correct if we were using the 5-kingdom system that we learned. The answer they're looking for there is eukarya. They were talking about the 3-domain system where they've taken Monera and broken it into archaea and bacteria, and then all other 4 kingdoms go into eukarya. But if you put Protista, I will give you half because I had so many people do it, and really you were thinking correctly. Connor, your hand was up, sweetheart.
No, I just said Protista, so it's...
Just take off half.
No, I said Protista, so it's eukarya.
Yep, it's eukarya.
Okay.
Yes, sir. Okay?
All right.
Yes, sir.
I put Protista and then I erased it out, and then I put eukaryotic.
That's close enough. It's not a spelling test. Okay. Now, each question is worth 5 points, but that includes all of the vocabulary words. Each one is 5 points. Okay, so you don't count number 1 and all of that is 5 points. Each one is worth 5 points. It'll always be that way. And on my tests, you never add up your grade. You always start from 100 and subtract off whatever you missed. If you missed a half, if you missed 2 halves, you missed 5. If you missed a half of just 1, you missed 2. So just take off 2 points. Yes, Audrey?
What was A again?
Heterotrophic? Was that the question? That was...
Because I had a heterotrophy.
Okay, but that was somebody that didn't make their own food. So you could put ate somebody else.
Can you put depend on others?
Depend on others for food? Yes.
Food somewhere else.
Yes, food from somewhere else? Yes, Geneva.
With number 8, if you got like one of those wrong, like the kingdom or something, what would that?
Well, that would be half because it's half. There's 2 answers there, so let's just take off half.
For F, I put organisms that can reproduce within their kind. I didn't put that their children could produce them.
Half. Just take off 2.
This,
Yes, honey.
I mean, I don't know if this is correct, but for the definition E for prokaryotic cell, I put a single-celled organism.
No.
I mean, I was just...
It's okay. Good guess. And you're right. Prokaryotic cells are always single-celled organisms, but there are other single-celled organisms. Okay?
Okay.
Okay. All right, so 5. If you got less than 80, I would suggest you restudy it and take it again because you're just getting used to his tests. Okay? And that's an option you have. Don't study the test to retake the test. Restudy the chapter in the study guide and then retake the test. Okay? All right, guys. Let's put that away and go to chapter 2. Yes, sir.
Okay, when you said in the notes what to prep for,
Right. There was apparently some confusion on that. You were just supposed to write the prep. You didn't have to actually...
2.1.
Mm-hmm.
Okay,
You didn't have to set it up yet because you're going to be doing that this week. Whenever it says prep for lab in your study guide, in your syllabus, it said not to write it? Then you're fine.
it says not to write 2 points.
You're going to need to write it up for this week as 2.2. So, in case you, I know there was some confusion on this. Everybody is supposed to get a sample of water from the scummiest source you can. Some of the best samples I've seen have come from fountains that are no longer functional, that are totally green, bird baths, dirty, scummy bird baths, really good place to get a sample. You don't want to get it from a place where there's running water. So some canals will work, some canals are way too clean. If you live in a community where the pond is aerated with a fountain, probably going to be way too clean. You're probably going to need to go find a drainage ditch or something that happens to have water in it. When you find your scummy sample, okay, you want to take just a tiny, tiny, tiny sample. We need a drop. So do not come in here with big jars. Just don't. If you've already set it up in big jars, go find little jars and pour a little into a little jar because I am not putting my hand down in these jars. And if you do, you'll stink all day too. So trust me when I tell you, you don't want to do that. This is the stinky lab. Everybody that'll walk by this room that has had biology will go, oh, it's bacteria lab day. Because it stinks. And everybody for the rest of the day are going to smell what we did in here first period. So I'm telling you this now. Okay? So what you want to do is get small jars. If you know somebody with a baby, I would suggest going and getting 4 baby food jars. That would be perfect. Okay, or I've had kids use the little tiny Tupperware type containers, the little tiny containers. You want the little ones. You only need a drop. And then what you're going to do is I would get a ladle to get your sample with. Don't necessarily stick your hand in it and use the jar. That's kind of gross. Get a ladle, and you want to scrape a little bit of the side. If it's a pond, you want to get a little bit of dirt. Not a lot, but a little bit of dirt in with your sample. If it's a fountain or a horse water trough or something, or a cow water trough, you want to scrape a little bit of the green off of the side and get it into your sample, okay? And then you're going to put, the book says to put hay in one. Most of you do not have access to hay unless you have bunnies or horses or goats. I don't think any of you do, though. But you do. Okay, there you go. Well, then if you have hay accessible to you, put a little bit of hay in there. If you don't have hay accessible, just get some dried grass. But try to get dried grass from someplace where it hasn't been sprayed with a bunch of pesticides, because then it's going to be counterproductive. Well, frequently they spray pesticides on the side of the road, so it depends.
I'm saying, like, I found a bale of hay.
Oh, there you go.
Used on the side of the road.
Okay, so there you go. But just do the best you can on that one. On the rice, it's specific that it's white rice. You can't use brown rice. Not sure why.
It has a shell.
Oh, there you go. So it's easier to access. That's good. So put the white rice in. Hard-boiled egg, you want to use just a tiny, tiny little bit of the yolk. And what was the fourth one? Oh, the dirt from the side. Okay, so you've got that one. Now the other thing is it says to keep it out of light. I'm going to tell you how to do this because they suggest brown glass, and most people don't have access to brown glass. So, especially not little bitty jars.
I do.
Okay, well that's good. I mean, because if you get supplements from your chiropractor, I've noticed they're all in little glass brown jars. And I thought, oh, look at this. But most people don't have that. So something I learned in cancer research, if you want something to be out of the light, put aluminum foil around the bottle. So all you do is grab a piece of aluminum foil, wrap the bottle in aluminum foil, and it is now without light. See, I learned that doing cancer research. That's what they did with everything. And then put it in your garage or your porch. But, big but, you must tell your parents and whole family that you're doing this so that somebody doesn't knock it over and it break, because it will stink to high heaven and everyone in your family will hate us forever. Okay? So you're going to want to make sure everybody's aware of it, that you put it in a place, maybe put it in a shoebox in one of these places so that if the shoebox gets kicked, they're not necessarily going to go shatter all over the floor type of thing. Okay? So put it someplace safe, make sure everybody knows about it, cover it with aluminum foil, bring it in next week. We only need a drop. Don't wear your favorite outfit in case you get any on you so it doesn't stink for the rest of your life. I suggested to some young ladies, just bring dad's old shirt or something, you know, so that you can cover yourself like you would a painting smock, so that you can cover yourself, so that if you get any on you, you can strip it off and still be around people the rest of the day. Yes, Audrey?
2 questions. What's a ladle?
What's a what?
Ladle.
A ladle.
A ladle.
Ladle is the thing that you dip soup out of a pot with. Oh, and after you use your mom's ladle, make sure that you wash it like 3 times and put it in the dishwasher.
I throw it away.
No, I wouldn't do that, because if you clean it, it'll be fine. But I've been ladling out of scummy duck ponds for so long that, you know, people used to watch me like, what is she doing? Because I'd be out there, yeah, this is like the best. And I don't live there anymore. I can't get those samples. That was in Plant City when I lived up there. I had this one duck pond and we had the best samples. You could find all sorts of cool stuff. So it's a scummy little duck pond. And that's what you need, is like a holding pond, and if there's like ducks or something in it, it makes it better, you know? So, yes. Did you have it? You said a second question?
Yeah. How much water do you put in exactly?
Oh, baby food jar. And it could be one of those little tiny baby food jars. Yeah, so small jar. Because once again, we have to get a sample out of it, so don't come in here. I had a kid come in this week, and he had these literal baby bottles full. And I'm like, I'm not putting my hand in there. Are you putting your hand in there? There ain't no way. I said, go home, pour about that much into a little tiny baby food jar. And he's like, okay. So, whoo. And warn everybody, okay? Did you have a question?
No.
Okay. Okay, so that's what you're doing for your lab. You're going to set up your lab so that we can look at those samples next week. And then he tells you to keep those samples for the following chapter, and I'm going to tell you to ditch those samples, and we'll get fresh samples when we want to look at the Protista kingdom critters and algae and stuff, because then they don't have to stink. It's the bacteria, the anaerobic bacteria, that are such stinkers. And you know, we'll get fresh samples when we want to see just pond scum, because that's more fun. Then you get more living things going on anyway, because you haven't killed them by putting so many bacteria in there. The 2 best samples I'm going to tell you right now are rice and the egg yolk. That's where we see the best stuff. And if it's not cloudy, and it's good that you already set it up. If it's not cloudy, you don't have much bacteria to look at. If it's cloudy, you're seeing the bacteria in there. And so it'll actually dissipate the light. So that's what you're looking for. Now, well, obviously your starting water is probably not going to be cloudy. It's just going to be scummy, right? So go for that. Okay? All right. Don't fall in. Use the ladle. Tell your mom what you're doing and your dad so you don't get in trouble. Tell your little sister, the dog, Yes.
I put mine in like a plastic bag because if someone dropped it on the ground, I didn't want it to break and go all over the ground.
That's fine.
Okay.
Yeah, because it's anaerobic. And once again, it should be in a warm place, not in the house. I'm assuming all of you use AC. So it should be in a warm place. It shouldn't be cooked, but a warm place. Okay, 80 to 100 degrees is where the bacteria like it the best. And it should be without light. So that's why I just put the aluminum foil around it, and then you don't have to worry because the light can't get into it. Okay? Page 37, Module 2, Kingdom Monera. So we are looking at prokaryotic cells. We're looking at bacteria in this chapter. And he mentions here that microbes, remember when I say microbes, I'm talking about anything that is too small to see with the naked eye, that microbes were actually unknown to people until the 1600s, and it says in 1670, I'm going to try to say his name, Anton van Leeuwenhoek, that's how you say it, at least that's what I've seen, how you say it, but he is the one that made the microscope, and he looked down under the microscope, and he actually saw a bunch of things living in water, and he called it little beasties, which I can appreciate. And so he's, look at all those little beasties in the water. And he actually drew pictures of it. It was quite cool. And so that's where we started to realize that there were living things that were microbial. And then my favorite microbiologist is Louis Pasteur, because he's really considered the father of bacteriology and did a lot of the work there, even though he was really a chemist. But anyway, so we're looking at prokaryotic cells, and he said that some biologists have renamed Monera let me say it right, prokaryota. And so if you ever hear that, that's the same as Monera. Once again, all prokaryotic cells have no membrane-bound organelles, and they are single-cellular organisms. You never have multicellular organisms made up of prokaryotic cells. So in general, we're talking about bacteria. Bacteria basically are found everywhere. And they're so small, their cells are so much smaller than our cells that he tells you that if you counted every cell in your body, we have more cells of bacteria on our body, even though we're clean people, in and on our body, than we do our own cells. Because they're very, very small. They're much smaller than our own cells. When we look under the microscope, you can see eukaryotic cells at 100 power. You can get a pretty good view of a eukaryotic cell. Prokaryotic cells, you've got to go to 400 power, and they still just look like, they're really small. So just to give you a heads up on that. Are all bacteria bad? No. What's some good things that bacteria can do for us?
Make bread.
Make what?
Bread.
Well, not bread. That's yeast, and that's not a bacteria. That's a eukaryotic cell, believe it or not.
Helps us make cheese.
Good. I don't know about you, but I love cheese. And without bacteria, we don't get all those cool cheeses. And I know a lot of kids are looking at me like, what do you mean there's bacteria in my cheese? Well, you know that Swiss cheese? You know all those little holes? That's where the little guys are respirating, and they're making little pockets of air. And then when you cut through it, it's okay, because that's actually what cheese is. Cheese, I heard on one program a chef say, cheese is milk and bugs in perfect harmony. And I thought that was so appropriate. And so cheeses are made from different microorganisms. That's actually what gives the different cheeses the different flavors. So not all bacteria are bad. What's another thing bacteria do for us? There's another one that's very important. Yes, Connor? Cleans laundry?
Yeah.
Well, there are actually some bacteria that are used for their eating power. And they use some of them. I hadn't really thought of laundry, but imagine they could. I know when they have oil spills that there are certain bacteria that eat petroleum products, and so they seed them into the oil spills to break down the oil, because some bacteria are able to break down things nothing else can eat and then make it back into things that other things can live around. So you're right that there are some pretty interesting industrial usage. But guys, there's some that live inside of us. Do you remember, Dominic, by any chance? What were you going to say, honey?
For the other bacteria, bacteria that decompose organisms.
Oh, I'm so with you. Bacteria decompose dead things, thank you God, or everything that's ever died would still be laying around. That's a gross thought. And then I had some boys on the other coast go, yeah, but they wouldn't be gross because they wouldn't be rotting. Yeah, but I wouldn't want dead things all around me. Yuck. Even if they didn't stink. This is girl time. Ew. Okay? Yes. Yes, absolutely. And you're smelling the microorganisms, and that's why a lot of cheeses have a very strong odor. Yes. Well, it's different. It's different bacteria. And actually, you can identify certain diseases by their smell. I know there's a disease that puppies can get, and I've seen it in guinea pigs, and it's called coccidia. And we had a guinea pig that we bought, and then it developed this smell, and then it died. And when I took it to the vet, they said that was coccidia. So then when I had a litter of puppies and I smelled that smell, I took it to the vet immediately and said, we've got coccidia. And they checked and they go, you're right, how did you know? And I go, the smell. So you can actually identify, just like you could identify cheeses from the smell, you can actually, if you're familiar with these different bacteria and what they smell like, and now we're talking pathogens, aren't we? Because we're talking about diseases. Gangrene. Have you ever watched a movie where, an old movie, where they're removing the bandages and they see that somebody's got gangrene? Have you ever seen a movie like that? And they always, in the movie, go like this. Because it's never just, oh no, it's because the smell almost knocks them out. Because gangrene stinks. Okay? And we're going to talk more about the anaerobes in a minute. One of you had your hand up? Yes?
Have you ever heard of Kalamazoo? It's a cheese.
I've heard
It's like
tiramisu, but I haven't.
Kalamazoo.
No.
It's like only allowed to be made in one place, or otherwise it's like illegal.
Okay.
And it's like this cheese that they cut the top off and like leave it outside for like maggots and stuff to get in it, and then they give it to people.
How do you know these things?
My sister.
Okay, never mind. We'll just let that one go. All right, he's got a big family. I just want you to know that. So lots of sisters to pull from, at least two to pull from. So lots of brothers too. Nice family though. I love your family. You know that. Okay, you guys, nobody came up with that the bacteria are in our gut and they synthesize vitamin K, B vitamins. This is why if you're on an antibiotic for any length of time, your mom starts giving you yogurt or acidophilus or some probiotics to try to rebuild the stuff in your gut, because without the bacteria in your gut, we're going to be very sick. We're going to be vitamin deficient, and the good bacteria in our gut actually fight the bacteria. So there are friends, you don't want to lose them, okay? So there are good bacteria. I just want you guys to understand that. And then there are very bad bacteria. And disease-causing organisms are called pathogens. And I want you to understand, not all pathogens are bacteria. There's athlete's foot, which is a fungus, and that's a pathogen. There's viruses that cause diseases, and they're pathogens. So any disease-causing organism is a pathogen. When I worked in cancer research, I worked for the Department of Comparative and Experimental Pathology, because pathology is the study of diseases. Okay, so I want you to get that word root there. So a pathogenic bacteria is a disease-causing bacteria. Let's turn to page 38, and let's look at the typical bacteria. Bacterium would be singular. I never think of them as being by themselves because they're always with groups. So I always say it as if it's plural. But we're going to look at a typical bacterium. Now that's like saying a typical person. How different can people be? Pygmies, Shaq, we can get all sorts of variations, right? And bacteria, it's similar. So we're going to look at some typical characteristics that can be found on certain bacteria, but I don't want you to think all bacteria look like this. It's just we need to learn the characteristics. It's kind of like if we were studying people, we'd say, okay, they got eyes and a nose and mouths and ears and, sometimes they have hair even. Anyway, okay, you get my drift. So let's go ahead, and you're going to need to know the functions of these. So first what I'm going to do is I want to go through the parts of this cell that are the same as our cells. So there are things you're going to have to remember throughout the course, because all cells have certain things in common. And the one is that they are surrounded by a membrane, and whenever you hear membrane, you should think skin. It's like a skin. But what is the name of this membrane that's given around a cell that holds the cell in? No. Look at your thing. Look at your picture. What is the membrane called? There's a word before the word membrane. Good. Plasma membrane. And I'm going to shorten it to PM for plasma membrane because it's just too long to write. The plasma membrane, our cells have plasma membranes too. Plant cells have plasma membranes. All cells that I know of have plasma membranes because it's the skin that holds everything in. And it's a very specific skin, and you and I are going to be very familiar with it soon. Actually, if you, let me see, if you turn a page, don't lose your place, but if you turn to page 178, on page 178, you have figure 6.7, which is the construction of the plasma membrane. Let me inform you, you will be intimately familiar with the plasma membrane by the time you get to module 6. And so right now we're just learning that's a cross section of it. If you cut it, that's what the plasma membrane would be made like. So I just want you to know, we will know more about it. But the plasma membrane is real important because it's made up of, he tells you, phospholipids and proteins, and you're going, what is that? Well, you'll know all that in chapter 5. We'll study those. It regulates what's coming in and out of the cell. I've always told my middle school students that the plasma membrane is like the watchtower and the city walls around the ancient cities in the Bible. Because remember, they had city walls to protect everybody inside from what was outside or vice versa. And then they had watchtowers to say who could go in and who could go out, the gates. And that's how our plasma membrane works. And that's how this plasma membrane works. Yes, Jonathan?
All right, so how do they exactly know what's inside a bacteria?
Because people have spent like 30 years figuring this stuff out. And they use electron microscopes, and they do long, slow experiments.
Well, I was wondering, is it like they're see-through or like...
Yeah, you're going to be able to see through them, but they're so small, it still takes incredible patience to learn the stuff. And actually, because my degree is in cell biology, when we get to chapter 6, I'm going to tell you how they discovered some of the things they do to give you a feel for what the research looks like. Because when I was in school, I graduated college in 1981, and when I was in school, they didn't have textbooks for my senior level courses. And all of my classes were using the same research for different things. And when I noticed that, I bumped my girlfriend, who is now a vet and an attorney. I bumped my girlfriend and I go, hey, they don't know any more than this. Have you noticed that? I go, they're all using the exact same stuff to prove different things. They don't know any more than this. And it was cute because when I started teaching this book and I got to module 6, I'm like, oh, they finally figured out what the Golgi body does. And my kids are all looking at me, my students, I go, you don't understand, when I was in college, they were still figuring it out. And then I told them how they did the experiments to figure it out, and everybody's like, because it's very long and very slow. I'm going to tell you about the flagellum there. The flagellum, they spent over 20 years figuring out the bacterial flagellum. 20 years of research to figure out the bacterial flagellum. And I'm going to tell you more about that in just a minute. Yes?
Is that their indirect evidence?
No, this is direct.
Okay.
Yeah. It's incredible. They use iodine and other radioactive isotopes, and they actually mark a protein with, or a substance that the creature can eat or absorb. If it's like these, they don't eat, they absorb things. And then they track them as they, you know how hard that is? And they have to do it at specific intervals. I worked in cancer research for 2 years, and one experiment would take like 8 months. One experiment, because you had to inject the mice, and then you had to, wait however long that you were injecting the mice, then you had to harvest them, then you had to take that means kill them, and then you had to take their bone marrow cells and you had to squirt them out, and then you had to culture their bone marrow cells, and then once you cultured their bone marrow cells, then you had to test all of the cells, because you're doing this sterilely, all of this, under tissue culture, and you have to test them all to see which one's making what you're looking for, the whole point of the experiment. And then when you find a well in these 96 well plates that are this big with 96 little wells in them, when you find a well that actually is making what you're looking for, then you have to go into production and you have to start cloning that cell. You'll understand all this more after I teach you through chapter 7. And then we had to do that, and then you did that for months and months. And then you froze them down in liquid nitrogens, because once you found one, you were praying that it didn't just go away. And then you started the main experiment. You see what I'm saying?
Yeah.
I mean, oh my goodness, these are just long, slow, patient things. Okay, and we'll get back to that. I'll tell you guys more about that as we go. Okay? But let's do, once again, what we all have in common. Plasma membrane. There's 4 things all cells have in common, and they're here, so we'll just do them first. The next thing is, what do all living things have in their cells? DNA. And in the case of a bacterium, it's actually a circle of DNA, but if you took a circle of string and you dropped it on the floor, it'd look kind of like that, wouldn't it? So the DNA, we all have the DNA. There is something that looks like pepper in your picture. What is that? Does anybody want to try to say what that is? Very good, and you said it correctly. I'm so proud of you. It is not ribosomes. It is ribosomes with a long I. Okay, and I know I probably don't pronounce everything correctly, but I am a cell biologist, and that one just kind of grates on me when I hear it incorrectly, so you have to forgive me. It's ribosomes. Hey, at least you can sound like you took biology, right? Okay, so ribosomes. Ribosomes are protein factories, and you're going to need to know that forever because this is actually, if you think of DNA like a recipe book, like a cookbook for life, it's got all the information to make you or your dog or a plant. The ribosomes are the kitchen where the cookbook is made into the meal. Does that make sense? So the ribosomes are actually where the information in the DNA is taken and made into whatever it's coded for. And so they are actually a non-membrane bound organelle that our cells also have. Okay, there's one other thing that all living things have. What is all of this suspended in? Cytoplasm. Cytoplasm. And I'm going to tell you right now, this word root, cyto, means cell. And you're going to want to remember that because we're going to learn about cytolysis and cytology and blah, 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 blah. You need to know cyto means cell. Okay? So cytoplasm is the jelly-like fluid that everything is suspended in. Our cells have a plasma membrane. We have DNA, we have ribosomes, and we have cytoplasm. The cytoplasm is full of all sorts of chemicals so that different reactions can go on there, and it's actually a very busy place. Okay, now from there we're going to go into some bacterial things. The next layer out, if it has another layer, and it doesn't necessarily, but if it has the next layer out, what is that? Cell wall. Thank you. Cell wall. I think it's dead. A cell wall. Now, plant cells have cell walls. Algae have cell walls. Some bacteria have cell walls. Not all of them. The cell wall actually gives them shape. And I want you to look on page 39, which is right across from where you are. Bacteria come in 3 shapes. They come in a circular shape called cocci. That's the plural, coccus or cocci. And circle and cocci start with a C. So even though they're spherical, it just makes it so it's easier to remember. Circle, cocci, okay? The bacillus or bacilli are actually, he says rod shape because bacilli or bacilla means rod. I'm going to tell you bacillus are bat shaped, like a baseball bat, because they both start with BA, right? Bacillus, bat shaped, okay? And then spirillum or spirilla are spiral shaped. Now a helix or helical is spiral shaped. I just want you to know that. So if he says helical, you'll know he's talking about this. It's a spiral shape, spirilla. Those are the 3 shapes that bacteria come in. If they are bacillus or bacilli, or if they are helical, they're spirillum or spirilla, then that means they have a cell wall. Because otherwise, if you don't have a cell wall and all you have is a plasma membrane, you're going to be a little round sphere, like a water drop. Okay, and I don't know if you know this, water drops only have that pointy shape because they're coming off of something. If it's in midair, mid-fall, it's circular. It's just a sphere. And so these guys without a cell wall are spherical. Just want you to know that. Okay, so the cell wall. Then if they have something on the outside of that, and only certain ones do, what do we call that?
Capsule.
Good. Some of them have a capsule. And the capsule actually has 2 jobs. Does anybody remember one of the things the capsule does?
It sticks.
Good. It sticks. It sticks to things. Now, I'm going to tell you one place that these are found. Between your teeth. This should gross you out. I'm hoping it does. Okay? All right, when these bacteria that are encapsulated bacteria get between our teeth and we brush our teeth, we only brush this surface and the back surface and this surface, but we don't get the surfaces between our teeth, which means we're missing 2 out of 5 sides. You got 5 sides to your tooth: front, bottom, back, and the 2 sides. Oh, that's why we're supposed to floss. We are supposed to be flossing to disrupt the little capsulated bacteria so that they are knocked off of the side of our tooth so they don't climb up under our gums and give us gingivitis and eat away at the root of our teeth. And we'll talk more about that when we study amoeba, because I'm going to gross you out totally, because I hope everybody starts flossing their teeth. Anyway, the other thing that the capsule does is that it protects them from disease-causing organisms. And so that's important for them, because we have antibodies and things that our body does to attack bad guys when they come into our body, and the capsule is supposed to protect them from that. And so this helps them to, so I would think that the capsules you would see in pathogenic bacteria, you might see it more in them. Okay? And let's see, what did I miss? Just the tail, right? So the last thing here is the flagellum. And the flagellum, woo, and it's not flagellum, it's flagellum, okay? And flagellum, like jelly. And in plural, it would be flagella. And so here, flagellum. And they can't have more than one. If they have it, they can have locomotion. I hope we see some flagellated bacteria next week, because if they go cruising across the screen, you'll know they're flagellated, because that's the only way they can do that. So that is locomotion. Jonathan, you had asked a question. They spent over 20 years, researchers, spent over 20 years figuring out the parts of the bacterial flagellum. And if I remember correctly, there were over 201 functioning parts. There's, I know, well over 100 functioning parts to the bacterial flagellum. And that was where the intelligent design movement started. From scientists that were not Christians, not Jewish, not believers in creation, but as they studied the bacterial flagellum, they saw that it was like an outboard motor. And they said, how could this have just happened by accidental random chance processes like mutation? It couldn't. You could not get an outboard motor to run if you just happened to put all the parts in one place and shake it up. It wouldn't work. Anybody that's ever been on a boat knows that. And so when they looked at it, they were honest enough to say there had to be some kind of design here. Otherwise, you shouldn't see this. And that's where the intelligent design movement, it's called irreducible complexity, that if you took away one piece of the flagellum, it wouldn't work anymore, and therefore it's irreducibly complex. And that's where the idea of intelligent design came in, just so you know. Okay, these guys are only a micron, or a micrometer, a micron, and that means, you know, most of us are going, okay, a meter, a millionth of a meter, I can't get my head around that. I just no picture at all. So he tells you that 1,000 bacteria could fit on the dot of an eye. That gives you kind of a feel, right? They're very small. You'll see next week. And these are supposed to be the simplest forms of life, and they're not simple at all. There are actually bacteria that can tell, they have little compasses in them, and if you turn them, they'll turn back. Yeah, so don't ask me how. Nobody quite knows, but they do. So, and we're going to see some more cool stuff that they do. They can reproduce themselves a generation time of 20 minutes. So that means from the time they divide until they're ready to divide again and do it, 20 minutes. So these guys can reproduce very, very quickly. Okay, on your owns, let's look. I'm going to read these to you just because we're running out of time, because we took time on the test. On page 41, 2.1, it says, different species of bacteria attempt to infect an organism. One bacteria succeeds while the other is destroyed by the organism's infection-fighting mechanisms. What is most likely the major difference between the two bacteria? What do you think is lacking on the one that's destroyed?
No capsule.
Good, and that's how he's going to ask questions of you to know that you know the functions of these. This is the way he'll do it. Number 2.2, a bacterium is poisoned by a substance that was allowed into the interior of the cell. What component of that bacteria didn't do its job?
The plasma membrane.
Good, plasma membrane, didn't keep it out. And if a bacteria can't move, what is he missing? Flagellum. That's the only way they're able to move. Okay, on page 41, we're given the word saprophyte. 80% of all bacteria are decomposers, but some of them are autotrophic. And some of them are consumers rather than decomposers. But 80% of all of them are decomposers, and we call them saprophytes because they live on dead tissue. Turn the page. On page 42, he tells you if they live on living tissue, they are parasites. So if they live on dead tissue, they're saprophytes. If they live on living tissue, they're parasites. If they're autotrophic in a bacteria within Monera, they can either be photosynthetic, which means they take light and make food out of it, but he tells us here that if they do that, they make different byproducts than plants do, but they're still useful byproducts. But they can also be chemosynthetic. Chemosynthetic means they take chemicals and break the chemicals apart and use the energy from breaking the chemicals apart to actually make their food. And he says that the chemosynthetic bacteria are able to take chemicals that other organisms can't use, like what I mentioned with the petroleum products. Most living things can't use that for food. They can take that and then convert those things into a food source. So the chemosynthetic bacteria are very important too. If an organism needs oxygen to survive, it's called aerobic, like aerobic exercise, right? Okay, aerobic, because they need oxygen to survive, and some bacteria are aerobic. They can live in the presence of oxygen. Some are anaerobic, and anaerobic bacteria cannot live in the presence of oxygen. So if you really want to kill them, you would oxygenate an area, expose it to the air. The most dangerous bacteria that I know of are anaerobic. Tetanus is an anaerobe, and that's why when you get a puncture wound, your mom pushes at it trying to get it to bleed, and you're like, leave it alone, it already hurts, and she's going, no, you have to make it bleed. Well, the reason you have to do that is because you can't wash out a puncture wound very well, but if you can make it bleed, the blood will wash it out, and you don't want to leave those anaerobes in there. If you expose them to the oxygen, they'll die, but if you leave them in there, you could end up with tetanus, gangrene, is an anaerobe. Remember I said, because it stinks so badly, and anaerobes stink. And I've got to tell this story even though we don't have much time because this is the story. When I was in my college micro class, in Mr. Wilt's class, and we had a lab. In the lab, there were about 100 people in the lab, and I was sat close to the front because I was always blind. I've had laser surgery since, but I was up here. And so there was a good distance between me and the back of the classroom. We were using a botulism bacteria that day, and that is very dangerous. The one that grows in cans, botulism, and that's why you never eat from a respirating can, a can that's swollen because something's breathing in there, that's why it's swollen. Okay. Like they say, like 2 grams of botulism toxin could wipe out the population of the world. Very, very deadly. So we were working with a botulism organism that day, and Mr. Wilt said if anybody doesn't use perfect aseptic technique, he was going to fail us because it was just so dangerous what we were working with. And then he gave this picture that I still remember the picture he gave because it was so ew. He said, when you open this, it's going to smell like a cow has been dead in the August sun for 2 weeks, and then somebody walks up and punctures its stomach. And I remember going, that is so gross. Like, how could he come up with that? That was just so gross. And then, I'll never forget this. Remember, I'm sitting in the front. Some kid at the far back end of the room opened, and we're talking little tiny test tubes that these things were in, opened it up, and the whole class went silent and turned around and looked and went, oh, no. I was like, oh, this is terrible. Next week will be bad, but it wasn't. I've never smelled anything like that. It was just awful. And that's why when I see those movies where they go, like this with the gangrene, I can relate because isn't it kind of God to put the anaerobes where we can't smell them? You know, they don't live in the presence of oxygen. They're the ones that are at the bottom of the septic tank, you know, in the bottom of the swamp. And then when we get a whiff, we're like, you know, so anyway, so anaerobic bacteria are the ones that live where there isn't an oxygen environment. Turn to page 44, please. And he mentions that biosphere didn't work because man didn't come up with all the things God came up with.
I went there.
Cool. On page, we don't have time though, and I'm sorry. On page 44, 2.4, it says, can saprophytic bacteria be autotrophic? No, because the saprophyte, remember, lives on dead things, and an autotroph makes its own food. Can an aerobic bacterium be chemosynthetic? Yes. Sure. Just means that they're in the presence of oxygen. All right, then we're going to look at the different forms of reproduction that we see in bacteria. The normal way that all bacteria divide is called binary fission, and that's what's pictured on page 45. They attach the DNA molecule, which I told you was circular in a bacterium, to the plasma membrane. The plasma membrane stretches out It pinches down in between, and now you have two copies of the exact same information. What type of reproduction is this, sexual or asexual? Asexual reproduction. You have no new information. It's exactly the same as the parent. Turn to page 46, please. They can do this every 20 minutes, and he points out that if they divide at this rate, that within 10 hours there would be more than 1 billion bacteria, and within a week, if they could grow unabated, they would actually outweigh the planet, because they exponentially grow. Thank you, Jesus. They can't do that, though, because they wouldn't have enough food. So, and I think it's interesting that the dead cow thing comes up here again, and that's exactly what my professor used. It must be a bacteriologist thing, the dead cow, okay? But they pointed out that if the bacteria were on the dead cow, that they would have a bacteria party. Woo, bacteria party, and they'd grow and grow and grow, like, woo, lots of food, And then eventually they'd get to a point where they couldn't grow like that. That's exponential growth because they're growing like crazy. And they have plenty of food. They finally start to run out of food and it levels off. And in your picture, it's got figure 2.5 where the curve, let's see, for you guys it's this way. Nope, it's that way. So the curve levels off, and that's called steady state. And it says there that that's when the population is dying as quickly as it's being born. Bacteria are not born. It's dying as quickly as they're coming into existence, okay? Or dividing. As quickly as they're dividing, they're dying. So one way or another, steady state is an equilibrium, okay? You have equal dying to coming into being. And then eventually, and that would be your logistic growth. So the exponential growth is growing out of control because they have lots of food. When they get here, that's logistic growth because the growth is controlled by limited resources. Okay? And so that's here, steady state. And then eventually, they run out of dead cow. They start eating each other. They start dying off. And that's the end of them. How would we get more? Give them more dead cow. That's how you get it started. This is why your mother wants you to clean up the kitchen before you go to bed at night. She doesn't want exponential growth going on in the kitchen. She doesn't even want logistic growth going on in the kitchen. Okay, so that's why we clean the stuff, so you're not feeding things. I know. Can you imagine being my child? These are the pictures I give my kids. It's like, you don't want to feed that stuff. Go clean that up. Okay, so 2.6. I'm on page 47. A population of bacteria grown from a single starter bacterium is rather fragile. When conditions are changed, the population dies quickly. Based on what you've just learned, develop a hypothesis for why this is the case. Well, each bacteria, we have bacteria that can live at the core of a nuclear reactor. We have bacteria that can live 20,000 feet above into the atmosphere on weather balloons. But even though they're tough, each of those bacteria only survive in one place. If you moved them to a normal place, they die. Okay? They're very specific as to where they're able to live in these weird places. And so because they can only clone themselves through binary fission, you don't get any new information, and therefore they're very fragile if you move them from one place to another. Does that make sense? Whereas with sexual reproduction, you have the possibility of getting the information from two parents. And so with sexual reproduction, there's more adaptability that you see within any organism. All right, 2.7, a population of bacteria reaches a steady state, and then after several days, the population actually increases dramatically. What could cause such an event? More dead cow, right? Okay, more dead cow. Turn over to page 48, please. The other forms of reproduction that he'll mention is really genetic recombination, because now you're not actually talking about, but he's going to ask you what are the different forms of reproduction, and you're supposed to list all these. But really, this is genetic recombination. Conjugation is a very interesting thing, and that's pictured in figure 2.6. Now, the definition given there is a temporary union of two organisms for the purpose of DNA transfer. First, you have to know what a plasmid is. The DNA of the bacteria that tells it to be a bacterium is its main DNA. They can also have little accessory DNAs, little circles of DNA, kind of like accessories on a car. You could have a car without it, but ooh, I got an accessory. I got a, fancy leather seats or whatever. Bacterium can have these also, bacteria, and these plasmids. So the plasmid's a little extra piece of DNA that gives it a little accessory. And so if the plasmid, let's say, helps it to survive in hot water, and the other one will die if it's in hot water, it can share that genetic information through conjugation, and that's pictured on figure 2.6. It'll actually make a copy of that DNA. It will grab hold. Oh, I forgot the pili. Shame on me. These guys have fimbria, which looks like he's having a bad hair day. Fimbriae is the plural. And they grab things with that. So here in conjugation, they actually grab the guy next to them that they're going to conjugate with. And conjugation is a donation process. One guy makes a copy and sends that, actually takes a half of his plasmid, sends it to the other guy, and then they both make it's kind of like making a picture from a negative. So he sends that through the conjugation tube, and then there's a donor, there's a recipient, and then they both make the other half, and now both of them have the accessory. Okay, that's interesting. People can't do that. Eukaryotic cells, I've never heard of a eukaryotic cell being able to do this. So they're able, and then once again, we have to question, how does the bacteria know he needs that accessory? Oh, no. Yes?
How fast does that take place? Is it like shorter
Yes,
than the
yes,
production?
yeah, it doesn't take long. Everything they do is pretty fast. Sad for us. Yes?
Bacteria went to the body shop and got his car customized.
Yeah, basically. That's it. That's it. So on page 49, on your own, 2.8, a population of bacteria are living in a lake. Due to volcanic activity nearby, the lake's temperature begins to increase. In the population, there are some bacteria that are resistant to low temperatures and another type resistant to high temperatures. Which type will be the donor and which will be, as they begin to conjugate, the donor will be the one resistant to high temperatures. Okay? Then he goes through transformation and transduction. Now, transformation... Read what it says. It says the transfer of DNA segment from a non-functional donor to that of a functional recipient. You know what it is? One guy dies, the bacteria dies, and he opens up, and the other bacteria goes, ooh, I like that accessory, and he comes and he grabs the DNA for it. How do they know? I don't know, but that's what it is. So I would define transformation as a salvage operation. That's how I define it. It's a salvage operation. You're going to get the stuff from the dead guy and use it. Okay? Isn't that a salvage operation? Yes. Kelly, do you have a question, sweetheart?
What's the difference between the conjugation and the asexual reproduction?
Oh, total. Asexual reproduction, you're actually getting 2 new organisms out of the original one. Conjugation is that I have an accessory for a AM/FM CD player, and I'm going to give you a copy, so now you'll have one too. You're not me. You're not exactly like me. It's like, thank you. It's like burning a CD for somebody, which is illegal. But it's like burning a CD for somebody and giving them a copy. Okay, I'm not going there. Okay, so conjugation is a donor-recipient situation. Binary fission, you're getting 2 individuals. Asexual reproduction. So these other ones are genetic recombination because you're getting new genetic possibilities, getting accessories through conjugation, getting accessories through scarfing it from a dead guy. Okay, that was transformation. Transduction is where actually a virus that actually transfers some bacterial DNA from one bacteria to another. And we'll study viruses later. Just trust me to remember that transduction is where the bacteria gets it from a virus that happens to infect it with other bacterial DNA. Okay? Then he mentions an endospore on page 50. And an endospore, he says it's okay. I know a lot of you guys don't watch sci-fi. I guess you got computers now so you don't have to. We used to watch a lot of sci-fi, Star Trek, stuff like that. And when people wanted to go great distances in the galaxies, they would put themselves into suspended animation so that they would live till the ship got there. That's kind of what an endospore is like. The bacteria, if it finds itself in a bad situation, can't survive, it will put hard coatings around the outside of it, and then it'll go into like a hibernation state. It's not really living, it's kind of suspended. And then when it gets through whatever that hard situation is and the food source comes back or the temperature gets right again or whatever, then it's able to start growing again. And it said that that's why when that one gentleman boiled the broth, he didn't boil it long enough, and the organisms had endospores, and so they just came back, whereas Louis Pasteur boiled the broth long enough that it killed things with the endospores. Maybe your mom's told you sometime on certain foods, you don't just warm them up, you make sure you cook them. You bring them to a certain temperature before you eat them. That's because of these kind of bacteria and these endospores. And then on page 51, we're told about colonies. Now, bacteria can make colonies. They still have the 3 shapes, and you notice you don't see any spirilla here in this shape. You don't see any spirally colonies. I don't even know if they do. But the DNA within them tells them what kind of colony to be. It's not that you can break one apart and it'll go start to be a colony with another shape. The DNA tells them what kind of colony to be, and these colonies benefit these microorganisms because they're actually able to hunt food together. It gives them protection, their safety in numbers type of thing, and that's true of a lot of microorganisms. And so we can identify what type of bacteria it is. Like if you have strep, the doctor will look under it and he'll see the line of them and he'll go, oh, you've got strep. So this is one of the ways that we can actually identify what kind of bacteria we're looking at. You do not have to memorize these. What you have to be able to do is if I said staphylococcus, you would know that coccus meant they were little balls. If I said Staphylobaccillus, you would know that I was talking about little bats, right? Okay? So that's what you have to know about it, and that the DNA actually codes for what type of colony they'll make. On the bottom of 51, 2.9 says a bacterial colony is called Staphylobaccillus. What shape do the bacteria in the colony possess? Well, bacillus, what shape is that? Rod-shaped or bat-shaped, the way you want to say it, whichever one. Okay, you're going to set up your colonies. Read the rest of the chapter, do the study guide, don't take the test yet. Yes, Kelly? 2.2. You combine 2.1 and 2.2. Have a good week, guys, a Jesus-filled week.